All right, this is my final presentation on the cereal weevil and Sodalus bacteria symbiotic system. I have titled this presentation, I Ran Out of Weevil Puns, because I ran out of weevil puns. These are harder to come up with than they seem. The host of this system is the grain weevil, also known as the cereal weevil. They are beetles in the genus Sitophilus. There are a variety of species of this beetle, each one specializing on a different type of grain. There's a weevil that specializes on rice, there's a weevil that specializes on corn, etc. They feed exclusively on grain, specifically the type of grain that they specialize on. They lay their eggs in the grain, and their larvae develop inside this dried grain kernel. Now, this diet is very rich in energy in the form of starch, but it tends to be very, very low in a variety of vital amino acids and minerals that the beetle requires for various life functions. The symbiont of this system are bacteria of the genus Sodalus. Much like the host weevil, there is no one species of bacteria involved in this system. It is a variety of species. They are gram-negative gamma proteobacteria, and they are in the Enterobacter family. They are considered a nutritional intracellular endosymbiont. So their function is that they supplement the nutrition of their host. They live within host cells instead of in the intracellular matrix. And of course, endosymbiont means they live inside of the host's body. This is a reasonably important system for study. Grain weevils are a very common agricultural pest. Each kernel of grain that they lay an egg in essentially becomes inedible to us, as the internal contents of that kernel are eaten by a developing beetle. We would not like that to happen. We would much rather be the ones who eat that grain. And the symbionts, as I will show throughout this presentation, are the only reason they are able to survive this incredibly limited lifestyle. If we are to find a means of preventing this from occurring and preserving our grain, we are going to need to study the weevil and, by extension, its symbionts in order to deal with them. The currency in this system is pretty reasonably simple. As I mentioned a couple of slides ago, this is a nutritional symbiosis, meaning that there are nutrients being exchanged. More specifically, the beetle is providing its symbiont with starches from its diet. Grain is very high in starch. And the Sodalus bacteria is providing its host with a handful of amino acids and vitamins that it can synthesize, but that the host cannot. In addition, the Sodalus bacteria is given a safe environment where they are reasonably free from competition with other bacteria. Different symbiotic systems have different means of transmitting the symbiont from parent to offspring, or in some cases from environment to offspring. Although in the case of this system, it is indeed parent to offspring. It is a form of vertical transmission in which symbionts are passed from parent to offspring in the germline cells produced by female cereal weevils. Essentially, the symbiont is already contained within the ovum before the offspring even begins developing. There are no behavioral means through which this has to occur. Each weevil is born with its symbionts already contained within its body. I will explain later that a variety of factors lead to this producing an incredibly potent genetic bottleneck every generation for these symbionts, because such a limited number of them are passed from parent to offspring, and there is no exchange with the environment in this system. Once the weevil larva begins to develop, the symbionts move to bacteriocyte cells. Those are specialized cells contained within an organ attached to the midgut known as the bacteriome. From there, the symbionts can receive starch that is brought in through the larva's diet and directly exchange that starch for for aromatic amino acids, as well as riboflavin and biotin, which are essential vitamins that the larva needs to properly develop. Within these bacteriocyte cells and the bacterium by extension, immune modulator genes have been observed to be very highly expressed. Insects do not have a specific immune system. Their immune system responds the same to all perceived invaders, meaning that they essentially have to switch off their immune system within these cells in order to prevent their symbionts from being attacked. Symbionts that are outside of the bacterium for whatever reason are still attacked by the symbiont, are still attacked by the immune system, and are not recognized as friends by the weevil's immune system. This is a figure pulled from one of the many sources I used to produce this presentation. It is a cross-section of the cereal weevil's anatomy. Specifically, it shows their digestive and reproductive systems. All of the areas highlighted in red are organs that are specialized for containing bacterial symbionts. So that would be the bacterium pouches attached to the midgut, as well as bacterioms attached to the female weevil's ovaries that allow it to pass symbionts from parent to offspring. Now, one of the reasons this system is so interesting is because of the incredibly strong influences the symbionts have over host development. In addition to triggering the formation of the bacteriome organs attached to the host's midgut, the nutritional supplementation provided by the symbionts is what allows the host to form its water-retaining outer cuticle while it develops into an adult weevil. This cuticle is essential for retaining moisture and, by extension, homeostasis in particularly arid environments, such as the inside of a grain silo. And the host cannot survive in the wild without this cuticle. It can be kept alive in a lab environment if it is kept very, very moist, but it will not survive under typical circumstances. Furthermore, once the host has reached maturity and it has a carapace and no longer needs this dietary supplementation, the buildup of amino acids in its body will eventually trigger the apoptosis or programmed cell death of the bacteriocyte cells that make up the bacteriome organ. I will explain later that this is catastrophic to the symbionts, but in short, it allows them to be attacked by the host's immune system. This is a rudimentary graphic I produced in BioRender that visually summarizes the effect that the symbionts have on host development. As you can see, on the left is a beetle that has its nutritional endosymbionts, and on the right is a weevil that has been rendered aposymbiotic, likely through the use of antibiotics, as I will discuss later. The aposymbiotic weevil is much more susceptible to water loss by evaporation, such as by the sun, or in any other arid climate. And the weevil on the right is, relatively speaking, protected from such environmental stresses. The weevil on the right, as a result, has died. I put little X's over its eyes to signify that it has died. This is a graph taken from another one of my sources. It demonstrates the level of relative water loss in both healthy and aposymbiotic weevils. Healthy weevils that retained their symbionts lose much less water and lose that water at a much slower rate than weevils that have been rendered aposymbiotic or without their symbionts through the use of antibiotics in a lab environment. Next, we're going to be discussing dysbiosis in the cereal weevil and Sodalis symbiotic system. My research on this particular topic was somewhat limited, as was all of my research on for this presentation, because most of the early research on this system was conducted in France, and as a result, all of the papers are written in French. I neither speak French nor trust machine translation to provide me with accurate information about scientific writing, so I just have to go off of what papers I could find that are written in English. On the topic of dysbiosis, I could not find any papers reporting on the existence of naturally occurring dysbiosis in this system, be it the result of an opportunistic infection or otherwise. I believe it is possible that the endosymbiont is far too degraded already to produce any sort of systemic infection in the host. The genetic bottleneck from generation to generation is so severe that I believe it is possible that the symbiont is hemorrhaging essential genes at an incredibly rapid rate. Additional studies have shown that exogenous infection also struggles to disrupt the host-symbiont relationship. So as I mentioned on the previous slide, this system appears to be somewhat resistant to exogenous infection. When subject to systemic infection from what is apparently a plant pathogen, Dideckia didanti, the host continued to downregulate its immune system within the bacteriome organs, specifically within the bacteriocyte cells. So even when the host was undergoing essentially an utter crisis in which immune intervention was all but necessary, the immune system did not turn on the symbionts contained within the specialized bacteriome organs, and dysbiosis did not occur. While I could not find evidence of naturally occurring dysbiosis, I did find a study in which a form of dysbiosis was induced in the host via the application of antibiotics. I found 2 studies, in fact. So when the host is dosed with specific antibiotics that render it aposymbiotic, essentially that wipe out all of its endosymbionts, the cuticle completely fails to form unless the weevil is kept incredibly moist in a very controlled environment. It simply does not survive to adulthood, and even if it does survive to adulthood, it is rendered less vital in all respects. All of its vital signs are much lower than that of a weevil that was able to develop normally. So although nothing in nature seems to be able to disrupt this relationship, we can still render a weevil aposymbiotic through the use of antibiotics. While this would not traditionally be considered a form of dysbiosis, as it is a part of the weevil's natural life cycle, I still felt it prudent to mention it here. Upon reaching adulthood, the weevil no longer needs supplementary nutrition to produce its carapace. Unlike crustaceans, which molt continuously throughout their lives, beetles, like all insects, have a finite number of molts before they reach an adult stage of life and cease molting altogether. When they stop molting, they no longer need to produce a carapace. This happening results in the supplemental amino acids provided by the symbionts building up in the host body, which eventually triggers an apoptosis of the bacteriocyte cells. The bacterial symbionts are spilled out into the midgut, where they are both attacked by the host's immune system and digested to reclaim some of the nutrients that have been stored within their cells. While this may seem cruel and treacherous and traitorous from the perspective of the symbiont, from the beetle's perspective, it is perfectly rational. As I said before, once they reach adulthood, they are no longer capable of adding new proteins to their carapace. They no longer have need of these symbionts, and yet they are still living in the weevil's stomach, feeding off of the nutrients that it is intaking. If the weevil did not remove the symbionts from its body, they would essentially become parasites. Of course, in the case of female weevils, a small number of symbionts are retained, specifically in the germline cells, so that they may be passed on to the weevil's offspring. For my first additional topic, I will be discussing how, while Sodalis is still the primary partner of the cereal weevil, they are still reliant on a variety of different nutritional symbionts that live within their digestive system, and that the specific makeup of their microbiome, much like in humans, can shift with their diet. The primary source for this subject was a paper that initially sought to analyze whether specific strains of wheat and barley were more resistant to consumption by weevils than others. The researchers hypothesized that yes, in fact, some would be more resistant. This was a study conducted on Sitophilus oryzae, also known as the rice weevil. It is a weevil that, as the name suggests, primarily feeds off of rice. The researchers' hypothesis was validated by the data they collected. Higher protein grains were shown to be more resistant to feeding by weevils than lower protein grains were. More interestingly, for the topic at hand, the researchers also conducted an analysis of the various experimental weevils' bacterial microbiomes, depending on which grains they had been fed on, and found that while Sodalis was always the single most dominant symbiont in every weevil, with at minimum 60% of the total bacteria in any given system being Sodalis, the weevil's diet did affect the remainder of the bacterial composition, showing that much like us, they have a variety of different symbionts that help them digest different aspects of their diet. This is a graph from the aforementioned study showing the bacterial composition of the gut microbiome of the weevils that were studied in comparison to the different types of grain that they were fed on. The top row, of course, represents the different experimental groups and different grain cultivars that were used, with the numbers themselves representing the percentage of the total bacterial community that each individual genus of bacteria made up. As you can see, in every system, the Sodalis bacteria were considered the eudominant taxa, meaning that they made up the vast majority of the total bacterial community. This table defines eudominant as making up over 10% of the total community. In every single one of these systems, the Sodalis genus made up well over 50% of the total bacterial community, with many systems having it be as high as 90%. The conclusions drawn from this study were that weevils are primarily reliant on their diet for starch and their symbionts for amino acid supplementation, and as a result do not subsist quite as well on higher protein grains. This in turn makes higher protein grains more resistant to weevil infestation. And the Sitophilus genus are aided by many symbionts that live within their guts and help them digest various nutrients, but their primary partners will always be Sodalis bacteria. My second additional topic was on a paper that measured various vital statistics of beetles that had been stripped of their symbionts and compared them to beetles that had retained their symbionts to demonstrate that having their Sodalis symbionts quite simply makes cereal weevils better at doing everything that they need to do. This was a study conducted on Sitophilus zeamaeus, a weevil that specializes on feeding on maize or corn. The researchers hypothesized that the symbiont load, that is to say the amount of symbionts contained within the weevil's midgut, may influence many biometrics of the adult weevil, such as size or respiration rate. Numerous behavioral traits of the weevil were also examined in the study. The researchers administered various different antibiotics to the grain weevils in order to control their symbionts, and they used qualitative PCR to determine the symbiont load after the antibiotics had been administered. essentially they were testing to see which antibiotics had the greatest effect on the symbiont load. As predicted, all observed behaviors by the weevils in this study were shown to be diminished in weevils with lower symbiont loads when compared to weevils with higher symbiont loads, except for resting time, which was shown to be much higher. And both the respiration rate and the body mass were lower in treated weevils than in untreated weevils. This is a figure from the paper comparing both untreated control beetles and beetles treated with a standard thermal treatment to remove their symbionts to beetles that had been treated with a variety of different antibiotics. As you can see, the behavioral metrics shown demonstrate that untreated weevils walk faster and are more adept at flying than weevils that had been treated to remove their symbionts, while the treated weevils were both slower at walking, worse at flying, and needed to rest more frequently and for longer. This paper has demonstrated that antibiotic treatments are more effective at diminishing the bacterial load of cereal weevils than standard heat treatment is, and that doing this renders them both less able to extract nutrients from their food, as well as generally less able to perform most basic life functions. This is believed to potentially reduce their ability to act as agricultural pests. Specifically, the antibiotic ciprofloxacin was shown to be most effective at eliminating the nutritional symbiont described in this paper. Unfortunately, that is an antibiotic that is used in case of emergencies in the face of multi-antibiotic resistant bacterial strains, and so should not be used willy-nilly to deal with agricultural pests. Even though antibiotics were shown to be more effective than standard heat treatment in this instance, it would be inadvisable to mass adopt them to deal with agricultural pests, as human agriculture is already one of the largest contributors to the production of multi-antibiotic resistant bacteria, and introducing more antibiotics into the equation is almost certainly a terrible idea. In summary, the Sitophilus cereal weevils depend on Sodalus bacteria living in a specialized midgut organ to supplement their nutrition. This supplemental nutrition is used to allow them to synthesize an outer cuticle, which allows them to retain moisture in arid environments, which is essential for maintenance of their homeostasis. This relationship is difficult to disrupt artificially, but can be broken up through the use of antibiotics, and must unfortunately come to an end as the weevil reaches maturity and no longer has need of its symbionts. That being said, I do not believe this is an example of what is often described as reverse parasitism, in which the host essentially parasitizes its symbionts. I do not believe this because if the host did not engage in this behavior, its symbionts would likely begin to parasitize it, as they would churn out amino acids it has no use for in exchange for starches that it desperately needs to produce offspring.